Hello everyone, this is Nandita standing before you to give you explanation about cell division. In this cell division, the phases of mitosis and meiosis. Students, if I gave a seed and I ask you to break down the seed into two pieces, you may observe a plant inside? No. In the meanwhile same, I gave an egg and I ask you to break down the egg into two pieces. Did you find a baby chick inside? No. But when we sow the seed in the soil and water the seed, we will get a plant and which this plant grows up to a tree also. In the same time, we will hatch an egg, we will get a baby chick inside. Students, have you ever thought how this process takes place? Let us see by this in this cell division and the phases of mitosis and meiosis, how the cells are divided, how the organs are formed and organ systems are formed. For example, take an examples of all the plants and animals make their beginning with a single cell called zygote. Zygote is formed by a eukaryotic cell after a fertilization event between two gametes. It is formed by a eukaryotic cell. In other words, zygote genome is a combination of DNA in each gamete and it contains all the genetic information which is necessary for a new individual. Here we are telling that zygote is an eukaryotic animal. So we have two types of cells. One is eukaryotic and another one is prokaryotic. Have you heard the word, students? What is a prokaryotic cell? And what is the difference between in these two cells? In eukaryotic cells, the nucleus is present, while in prokaryotic cell, the nucleus is absent. These are the major difference between prokaryotic and eukaryotic. For example, bacteria is a prokaryotic cell because in bacteria the nucleus is absent. So now we are discussing about a plant or animal which begins their life with a single cell called as zygote. This zygotes from the cell division, it divides into 2 to 4, 4 to 8, 8 to several thousands of times. This from single cell to several thousands of cells together form as a tissue. A group of tissues together form as an organ. A group of organ together form as an organ system. A group of organ systems together form as an organism. Let us see how this process are taking and we will start first from the cell division. The cell produces two types of chromosomes. The cell produces from egg or a zygote are two types. There are stomatic or vegetative cells or germs or reproductive cell. For example, the cell which produces an egg or a zygote we can call as stomatic or vegetative cells, germs or reproductive cells. The various organs in these organisms are derived from these two types of cells. Let us see in plants. From the plants, root, stem, leaves, etc. are derived from stomatic cells. Comes to the germ cells, pollen mother cells, megaspore mother cells, these are derived from germ cells. Now let us come to the animals. In animals, skin, bones, lungs, kidney, muscles, brain, etc. are formed from the stomatic cells. Testis and ovum means ovaries are formed from the germ cells. All the stomatic cells undergoes the cell division by the process of mitosis. All the germ cells undergoes the cell division by the process of meiosis. So the major difference of mitosis and meiosis is the number of chromosomes in daughter cell. In mitosis, the daughter cell will have equal number of chromosomes. When it comes to the meiosis, the daughter cell have exactly half of the number of the chromosomes in daughter cells. In a cell, the chromosome with two sets are called diploid cell. A cell with a single set of cell chromosomes are called as haploid cell. For example, this mitosis arc we can call as diploid cell, and the daughter cells of meiosis we can call as haploid cells. These are the major difference between mitosis and meiosis we can see in daughter cells. Now let us see about mitosis. Mitosis, it is a stomatic cell. The daughter cell resembles mother cell in each and every aspect in mitosis. The organs that are formed in this cell division such as skin, bone, lungs, kidneys, etc. These cells we can call as vegetative cells also. These cells, they do not directly participate in the reproductive system. The cells grown in any part means any system of plant and organism is due to the increase of the number of cells by mitosis process. The cells of younger and tender ones such as root, shoot and shrubs and tissues of young ones divides rapidly more than the cells from old and mature one. This is a reason for a great race of growth in organs and organ system. And before the division of mitosis in the cell, there is a various changes are seen. The major change in the cell of mitosis is the doubling of chromosomes number. After this completed, the cell divides so that the daughter cell is having equal number of chromosomes as the mother cell. You have already known that the chromosomes carries the genes. Genes carries the genetic information and they are essential for the inheritance of parental features. In other words, genes are responsible for the reducing and reconducting and directing all functions in the cell along with the proteins. Children, have you imagined what is happening if the genes chromosomes are not double in the cell? So what is happening in the daughter cell? So automatically the chromosomes numbers will become reduced into half after a few divisions. Totally the chromosomes are disappear from the cell. So finally the cell leads to abnormal and it goes into death cell or death organism. So it is must be clear there must be a equal number of chromosomes in each cell, otherwise the cell goes into abnormal cell or death cell. So let us see the cell division in mitosis. Before the cell division begins, the uncondensed DNA or unfolded DNA is in the rest form of, and we can call this phase is interphase. In this interphase, the DNA duplication is happen and it is in condensation form. We can call them as chromatin. Nuclear envelopes, nucleus, all are there in rest in peace, and this interphase is increases the genetic material of the cell in interphase only. From this interphase, the chromatin starts condense and fit to form the chromosomes. After completing this stage, the cell enters into prophase. This is the first stage of cell division. So during prophase, the chromosomes are comes in decondensed state. The chromosomes are comes in decondensed state. So as a result, each chromosome means as a result, the shape of the chromosome becomes definite and distinct. So each chromosome contains two arms called chromatids. Two arms. These two arms are called chromatids. These two chromatids join together by centromere. The nucleus and the remaining part of the nucleus envelope will slowly starts disappear, and whatever the centrioles are there, if two centrioles are present in the cell, they starts gradually moves to the end of the cell, and from the each centriole a fine thread like of fiber starts appears. These fibers we can call as spindle fibers. Spindle fibers. A centriole with spindle fibers we can call as aster. Now this spindle fibers grows towards the center of the cell. This process we can see in the stage of prophase. Now let us see what are the changes we can see in metaphase. The second stage of cell division is called metaphase. In this metaphase, the chromosomes now move to the center of the cells and they arrange themselves in a row. This looks like a plate and we can call it as equatorial plate. This we can call as equatorial plate. Some of the spindle fibers are attached to the chromosomes to join to the centromeres. The rest of the spindle fibers moves towards the opposite sides of the centrioles to join to the centromere. When this takes place, completely the nucleus and other parts are disappear in this stage. Now after completing this stage, next we will see the anaphase. Another stage, this is the fourth stage of cell division, we can call as anaphase. Anaphase is the third stage of cell division. In anaphase, what is happening? The spindle fiber starts pulling the chromosomes towards the end of the cell. So by this, as a result, what is happening? The centromere splits into half and they pull apart along with the chromatids. So as a result, what will happen? The chromosome splits into half of the original members and half of the original members of the chromosomes moves to one side and half of the original number of chromosomes moves to the upper side. When these changes are completed, the cell enter into the telophase. This is the fourth stage of cell division. In this stage, the chromosomes are completely stops their movements and they are arranged at the end of each cell. So now in this stage, the nucleus membrane and the other parts of the nucleus structures starts to reappear, and these chromosomes, they are going into a decondensation state by the end of the nucleus. So what is happening? Two nuclei are formed, one on each side of the cell. So spindle fibers are gradually totally disappear. These are the changes we are seeing in telophase. After completing this telophase, the last stage of mitosis is called cytokinesis. In this cytokinesis, a new membrane is formed nearby the equatorial plate. This membrane we can call as cytoplasm. In plants, the cell wall is formed to divide the cell into two daughter cells. By the end of the cytokinesis, the cell divides into two identical daughter cells. Those all the first four stages means prophase, metaphase, anaphase, telophase together called as karyokinesis because they shows the division of the nucleus. The last stage of the mitosis division we can call as cytokinesis because only cytoplasm is a component that undergoes the function of this phase. From this, the mitosis division is completed and it gives two diploid daughter cells. Now let us see what is meiosis. We already know that meiosis or reduction division that occurs from gametes and they produce, that occurs from germ cells and they produce gametes. We discussed that the major difference between mitosis and meiosis is the number of chromosomes in daughter cell. The meiosis division from the cell the meiosis division by the cell of the formation is lead to the production of male and female gametes. Each cell is having male and female gametes. When the fertilization takes place, these gametes fuse to form a zygote. These zygotes consist two sets of chromosomes. Two set of chromosomes. One is from male parent through sperms, and another one is from female parent through ovum. Various organs are increased by this repeated division by mitosis and meiosis through this cell division. Let us see about it. Let us discuss about a detailed structure in meiosis. In meiosis, the cell division means the nucleus will divide it into twice. Then it goes onto meiosis 1 and meiosis 2. In meiosis 1, the daughter cells receives only half of the number of the cells from the mother cell, and in meiosis 2, it forms immediately after meiosis 1. It goes into a simple mitotic cell division. In meiosis 1, the daughter cells again regenerate, means second generation. It is produced 4 haploid cells, means one set of chromosomes, and at the end of meiosis cell division. By the changes occurs, meiosis also goes into 5 stages, that is prophase 1, metaphase 1, anaphase 1, telophase 1, cytokinesis 1. To understand these changes, again this prophase is going to divide it into 5 stages. They are called liptotin, zygotin, pakitin, diplotin and diakinesis. Chromatin in nucleus of mother cells is condensed and it is ready to form as chromosomes. Following this, each chromosomes means a segment of chromosomes are ready to form a pair of chromosomes. So one pair from mother chromosome and one pair of chromosomes from the father. So after this chromosome starts synapsis. Synapsis is nothing but to pairing of a homologous chromosomes. After completing this chromosomes synapsis, the chromosomes looks like an X. Means the chromosomes shows the crossing over. So the chromosome shows the crossing over function. After completing this, the chromosomes now it enters into the middle of the cell at the stage of diakinesis. After completing this diakinesis, the meiosis enters into metaphase 1. Metaphase 1. In metaphase 1, what is happening means the chromosomes are in the center of the cell and it looks like a plate, we can call as equatorial plate. The spindle fibers are attached to the centromere and they move towards the centrioles, and some of the spindle fibers are attached. The nucleus and the remaining rest of the nuclear structures will totally disappear. So here what will happen, exactly the 4 chromosomes, they are arranged in a line of equatorial plate. After completing this, now meiosis enters into the stage of anaphase. Anaphase 1. In meiosis, when anaphase stage, what happens, the spindle fibers, they start reduce the length of the structure. So the half of the number of the chromosomes will move to the end of each of the cell. So this is the difference between mitosis and meiosis. Once you compare to the anaphase stage in mitosis, what is here, the chromosome, the centromere is splits into 2 parts. So the equal number of chromosomes are passing at one side and the equal number of chromosomes will pass at the another side. Once comes to the meiosis stage, here the number of chromosomes means the equal size of the chromosomes is dividing. For example, 8 is there, means 4 is dividing this side and 4 is dividing that side. This is the changes we can observe anaphase in mitosis and anaphase in meiosis. Now let us see the stage of telophase in meiosis. In telophase, total the spindle fibers are disappear, the nucleus membrane and the nucleus envelopes are reappears, and the chromosomes again they coil to form a chromatin. A layer means a cytokinesis layer. It divides the cell into two daughter cells. Means these two daughter cells we can call as haploid cells. Haploid cells means a cell with one set of chromosomes. Diploid cells means a cell with two set of chromosomes. So by this stage the meiosis 1 is completed. After this, immediately this haploid cells goes into second generation, means again the nucleus is divides, and this haploid cells will act as parental cell for the meiosis 2 cell division. Let us see what are the changes that takes place in meiosis 2 cell division. Meiosis 2 cell division resembles has mitotic cell division. Same the process, whatever the process takes place in mitosis in prophase, metaphase, anaphase and telophase, same the process are taken by meiosis 2 in prophase, meiosis 2 in prophase 2, metaphase 2, anaphase 2 and telophase 2. The major difference is before going to the cell division, the haploid cells means the DNA is replicated, means doubling of the chromosomes. After doubling of the chromosomes, these cells starts dividing and finally they produce four haploid cells with unique. Four haploid cells with unique. From these, the division of mitosis and meiosis are completed. Now we will see what is the conclusion from mitosis and meiosis. From mitosis and meiosis, we have observed mainly the 6 differences. First one is number of chromosomes. So here in mitosis it is have equal number of chromosomes. Here half of the number of chromosomes. Now from comes to the DNA duplication in both cells are here in mitosis DNA duplication is there, in meiosis DNA replication is there. Now next come to the synapses. In mitosis synapses is not there, but in meiosis synapses process is there. Now come to the daughter cells. In mitosis daughter cells have equal number. So already I wrote there. So let us see the division. Number of division. In mitosis only one division is there. In meiosis there is 2 divisions. Next one is the doubling of chromosomes. In meiosis the chromosomes will doubling before the cell division. In meiosis the chromosomes will double after the meiosis 1. And last and final, the functions of the cell. In mitosis, the cell function are done with stomatic cells. In meiosis, the cell functions is done by germ cells. These are the 6 differences we observe in the process of mitosis and meiosis. And finally, the mitosis gives to 2 identical daughter cells with the same number of chromosomes. At the end of the meiosis, it gives 4 haploid cells with the one set of chromosomes. Children, from this you can learn how the cell will divides and how the cell divides takes place in from 1 cell to 4 cells, 4 cells to 8 cells, 8 to several cells, and these several cells together form as a tissue, tissues to organ, organs to organ system, and organ system to totally organization. First I gave an example as a seed or egg. It is a single cell. From now I think you can understand from that single cell how all this process will take place, and meanwhile that so many systems also included in organism. All this makes and all this cell division makes an organism. Thank you.